My name is Clara and welcome to Clarification. Today we're going to be talking about genetics. So before we get started, we just have to get through some basic vocabulary. Now I've put up all the textbook definitions for the things I want to talk about here. You can pause the video and watch here and just read the definitions, but if you keep watching, we'll just work through an example and really look at what each of these things means. So first, what is a gene? Well, if you look at our chromosome over here, which is just a tightly wound piece of DNA, and then you focus on a specific part of that DNA, that is going to be a gene. Now, it's not just any random part of DNA. It has to be a coding region, and that's what we call a gene. So this segment of DNA is coding region, which means it's going to be transcribed into mRNA and then translated into a sequence of amino acids or a polypeptide, which will then fold into its 3D shape and result in a protein. And that protein is going to do something, and then that is going to give us some sort of observable trait. So a gene is a segment of DNA that codes for a protein, and that protein does something, and then those are the traits that we observe. So the example that we're going to be looking at is, of course, you guessed it, peas. Of course, Mendel started studying genetics there, so we might as well start there too. So the gene that we're going to be talking about is actually 3,550 nucleotides long, and then eventually that becomes expressed as a polypeptide of 960 amino acids, and that makes this protein called SBE1, or starch binding enzyme 1. So this is an enzyme, which means it's going to catalyze a reaction within a living organism, and then what it does is going to end up eventually give us our visible trait, and in this case it's texture. So for peas, that could be either smooth or wrinkled. So how does this protein result in this trait that we can see? Well, this SBE1, what it does is takes this soluble starch, or amylose, and then changes it into insoluble starch, or amylopectin. And what it does is, since it is insoluble, when the water is going to leave the peas, it's not going to take the starch with it, and then this amylopectin is going to give the peas a nice structure, leaving them nice and round, not like all crumpled up and wrinkly. So we mentioned that we can have either smooth or wrinkled peas, but how do we get these two different versions of these two different textures? So that brings us to alleles. So alleles are just different versions of the same gene. Now you've probably heard this a lot of times. What does this actually mean? So a gene, as we said, is just a bunch of nucleotides that code for a protein, right? So when we say it's the same gene, in this example, we are talking about the gene that codes for SBE1. So there are different versions of this same gene that code for slightly different versions of this enzyme. So there's the normal gene that we already talked about, the normal allele, which is 3,550 nucleotides long, and it makes our normal SBE1, which does a job converting amylose into amylopectin, giving us brown peas. Now we have this other allele, which in this case is actually a mutation, and what it is it's the normal 3,550 nucleotides, but it also has an extra 800 nucleotides just inserted in the middle of this coding region of DNA, or this gene, which results into a different sequence of amino acids in our polypeptide, and then that gets folded differently, and it's still the same protein, so it's still the same gene coding for the same thing, but it's not going to work this time, so you're going to end up with an inactive SBE1 enzyme, and then that is not going to be able to convert the soluble starch, or amylose, into insoluble starch, or amylopectin. So you just stay with this amylose, and since that's soluble, when water starts to leave your peas as they mature, it's going to take that starch with it, and then the pea kind of loses its structure. It doesn't have anything to hold it into a nice round shape, and that's how you end up with wrinkled peas. So an allele is just a different version of the same gene that codes for the same thing, except it gives you like variations in whatever it's coding for, and then that results in a different trait. We can represent this smooth texture with a capital R and the wrinkled texture with a lowercase r. So that's because the uppercase represents that this trait is dominant and the lowercase represents that this one is recessive. I'll get into what exactly that means in just a moment. So peas, just like us, are diploid organisms, which means they have 2 copies of each chromosome. So if we look at this one chromosome here that had this SBE1 gene on it, they're actually going to have 2 copies of this chromosome, which means they're going to have 2 copies of this gene. Now, each of those copies can have a different version of that gene or a different allele, right? So we can have both of those chromosomes having the same allele, the same version of that gene. So that's what we call a homozygous genotype. So genotype is just showing whatever alleles you have present in this organism. So our genotype could be capital R, which is homozygous dominant, and we can have little r, which means it's homozygous recessive. So that is our genotype. And then our phenotype, well, if it's big R and big R, it means it's going to make the SBE1 protein without any problems. And then that is going to result in a smooth trait. Now, if we have the wrinkled, it means both of those copies have the gene for the inactive SBE1, which is not going to make, which is not going to be able to convert amylose to amylopectin, and then you end up with wrinkled peas. So that is our phenotype. So genotype is just saying what kind of alleles you have, and then phenotype is saying what it's going to look like. So our trait was texture, and the phenotype is what we'd actually see, so smooth or wrinkled. Now, what would happen if we have a heterozygous organism, which is to say that an organism that has 2 different alleles? So one copy on one chromosome is capital R, which is our active enzyme, and then the other one is little r. So that one has the mutation, and that's going to make an inactive enzyme. What would this P look like? So this chromosome has the capital R allele, this chromosome has little r allele. So this is going to make inactive SB1 useless, I don't care. This is going to make active SB1. So this P is still making some useful SBE1, which is actually going to go and convert amylose into amylopectin. And then, like, it might not be as much of this enzyme that you would have, like, say, in a homozygous dominant P, but this P still has some. And this enzyme is actually going to go and, like, do some work, and then you're going to still end up with a smooth, observable trait. So the genotype for this P was capital R, followed by a little r, and that is our heterozygote, and then we have the phenotype was a smooth P. So how did we figure out that smooth is dominant over recessive? Well, if you remember, like, the beginning of this video, the definition of a dominant allele is an allele whose phenotype is expressed or observable in a heterozygote. So this is our heterozygote, which is why we were looking at it, right? And then if you see the big R, its phenotype is smooth. Now, is this expressed in our heterozygote? Yes, it is. Therefore, smooth is dominant. Now, if you look at our little r, that allele's phenotype is wrinkled. Is that expressed in our heterozygote? No. Therefore, it is recessive. So smooth is dominant over recessive. All right, thank you for watching and learning about intro genetics vocabulary with me. If you have any questions, please leave them below. I'd be happy to clarify